All right, hello class. This is the second lecture on the urinary system, and I'm going to share my screen and put up my PowerPoint slides. There we go. All right, last time we had gone through an introduction to the urinary system, and we had talked a little bit about the organs that are involved, the kidney, the ureters, the bladder, the urethra, and we had gone through the organization of the kidney, the macro structure of the kidney, and then the major vessels and the ureters that are coming in and out. Talk about the blood flow into the kidney and into the nephron. We've been going through some of the microstructure of the nephron. Let me choose the laser pointer thingy here. Okay, so this is the nephron. We start with the glomerulus, then we go to the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. And then we have the collecting duct. And this is a zoomed in look at the glomerulus. We have the incoming blood through the afferent arteriole, goes through a series of capillaries here inside of the renal corpuscle. And then we have the outgoing blood. Okay, the blood that has been filtered is leaving. Okay, and that's blood actually continues to, even after it exits the glomerulus here, it continues to wrap around the tubule. They're called the peritubular capillaries. And there's a continued interplay between filtrate and the plasma, okay, the blood and the filtrate. So at this point where the blood is entering into the glomerulus, it gets filtered. That is, some of this fluid leaves the blood and goes into this space. It's called Bowman's space. Okay. And when it enters into Bowman's space, it will then go this direction to the proximal convoluted tubule. At this point, we call it filtrate. Filtrate is a fluid that is on the way to becoming urine. Now, we will not call it urine until the filtrate leaves the kidney and enters the ureter. As it enters the ureter and goes down into the bladder, then we call it urine. But until that point, we call it filtrate. Okay? So plasma's in blood, in the blood vessels, filtrate is in the tubule. Okay, that's important. These cells that are surrounding, they look like kind of octopus. They have little fingers or these interdigitated projections of these cells. These are called podocytes, okay? Podocytes cover the capillary and they function in filtration. Right. So we looked at a cross section here. These mesangial cells kind of help to hold together some of those capillaries in the glomerulus. And there's some regulatory activities of these as well that we're not going to get into. All right, so filtration happens in the glomerulus. Okay, filtration is the movement of fluid from the blood into the Bowman space. It's the initial movement of fluid that on the way towards becoming urine, the urine formation. Okay, it's a passive process, does not require ATP. The motive force here is blood pressure. The force of the blood in the glomerulus drives or powers or forces the filtration. So it's really the heart is powering filtration here because the heart produces cardiac output and the blood pressure. All right. We will talk about processes in the proximal convoluted tubule, in the loop of Henle, and in the distal convoluted tubule. Right now, it's pointing out the distal convoluted tubule, the area highlighted in yellow here. So we have active secretion of ions, acids, drugs, and toxins. We have selective reabsorption of sodium and calcium ions, as well as water that's happening in the distal convoluted tubule. In just a little bit, we will see how this position of the DCT is actually positioned over here, okay, closer to the entry of the blood into the glomerulus at the afferent and efferent arterioles. In this diagram, the diagram of the nephron, we have it conveniently laid out from left to right. The glomerulus, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and then the collecting duct all the way on the right. Okay, from left to right, it's a nice, easy diagram. But in actuality, all of this is kind of folded on itself, and the distal convoluted tubule here is actually right up close to the glomerulus. Okay, and so we only show this way to show the progression of the tubule, but in actuality, it's kind of jumbled together a little bit more than it is displayed. And in this way, we now understand that this tubule here on the left is the distal convoluted tubule because it's kind of been wrapped around. It's actually really close to this complex called the juxtaglomerular complex. These are a set of regulatory cells that are sensing stimuli from the distal convoluted tubule and then communicating that to the this juxtaglomerular complex to regulate blood flow. So you can think about it as a negative feedback or some type of feedback loop. Whatever we sense in the distal convoluted tubule, the filtrate has passed through the tubule, and we can then make some judgment. How are we doing? How are we doing in the nephron with doing filtration and reabsorption and secretion? And we can report back to the beginning of the nephron, in the glomerulus, and regulate how much blood flow is entering. Okay, so if it's too fast, if it's too slow, if we're getting too much sodium, if we're getting too many ions, too much water, then we can communicate back, say, okay, hold up, slow down, or speed up. And there's some nice feedback loop between the end of the line and the beginning of the line. All right, that's the role of the juxtaglomerular complex. Okay, these are the macula densa cells, which are a specialized type of these cuboidal cells that are on the distal convoluted tubule. So these are the cells there, the macula densa and the juxtaglomerular cells, which are between the tubule and the afferent and efferent arterioles. Okay, this endocrine structure secretes erythropoietin and renin, okay, or renin. So these are two hormones that we've talked about before. These cells are the cells that are secreting these hormones, EPO, erythropoietin, and renin. Okay, and these are hormones that have wide effects in other parts of the body, besides just the local control of the that nephron. So some of these are broad effects, some of them are local effects. So the goal of urine production here is maintain blood homeostasis. That is the volume and composition of the blood, and including the excretion of metabolic waste. These are the three major functions of the urinary system. Control the volume of blood and control the contents or composition of blood, as well as get rid of nitrogenous waste. Okay, again, here's one of these pathways. Over here would be endogenous protein versus dietary protein. When we think about eating protein, like eating like a steak or some other like tofu, this is dietary protein. So it's entering our body from our GI tract. Okay, but we also have endogenous protein, meaning we have muscle breakdown. We have other parts of our body that are continually breaking down muscle all the time. It's not just dietary, it's also internal proteins that are breaking down and finding their way into the blood supply. So those amino acids break down, and part of that amino acid breakdown includes the release of the deamination, okay, the release of ammonia from an amine, and this ammonia is toxic, so we got to get rid of that. And in the urea cycle, we studied that the liver converts ammonia into urea. Urea can be excreted by the kidney. Okay, so urea cycle happens in the liver, creates the urea. Urea travels from the liver to the kidney, and the kidney can release that. Right. So, what are the three primary nitrogenous wastes that are released from the released in the urine? Okay, urea, creatinine, and uric acid. Okay, and there are pictures of them here on the right-hand side, and in blue, you'll notice the nitrogens. Okay, that's kind of common chemical annotation to show oxygen in red and nitrogen in blue. Those nitrogens are the problem child, as I like to call them. Okay, urea is a product of ammonia waste that has gone through the urea cycle. Creatinine is a product of muscle decomposition from muscle tissue, so that's a waste product from muscle. And uric acid is another common form of nitrogen waste. It's just another unique combination of ammonia, and the way that urea can then be further metabolized into this other form called uric acid. Okay, so urea, creatinine, and uric acid, these are three most common forms of nitrogen in urine. Okay, they're dissolved in the bloodstream. They're therefore excreted readily in the urine. And so creatinine, just a reminder how the role of creatinine. Creatine is the good thing that muscles use to temporarily store a phosphate. Okay, they can transfer a phosphate from ATP onto creatinine for a temporary storage. It's like a ATP battery. We can temporarily store that phosphate, give it to ADP, create ATP in the moment, and transfer that into the muscular energetics. So we're going to probably give it to a myosin head during muscle contraction. So creatine is a valuable molecule for muscle activity because it's a temporary energy storage for muscle activity. But after creatine is used, it turns into creatinine. So creatine gets consumed, turns into creatinine. Creatinine is the waste product of creatine. All right, so you might have heard like workout supplements that has this, workout powder or drink or whatever has creatine in it. You take that, you're going to be able to work out better because it's supplying the one of the molecules that your muscles can use to store ATP. So it's not really giving you energy, but it is a way of helping you have all of the supplies that you need to do a workout. Okay? The effect is you'll feel like you have more energy to do your workout. The creatinine is the waste product. Okay, the creatinine is a nitrogenous waste that has to be it's produced in the muscle, flows through the blood, and arrives in the kidney, and that's where we release it as a waste. Okay, we put it into the urine. All right, so the kidneys usually produce concentrated urine. That is, the concentrations in the blood versus in the urine are going to be very different because urine is a concentration of those waste products. So it's going to be much higher concentration in the urine than in the blood. Okay, and so the milliosmoles here is this unit MOSM per liter, milliosmoles per liter, about 1,200 to 1,400 milliosmoles is the concentration. These are there's a way of counting the number of ions in a fluid. Okay, all you have to know is about 4 times the plasma concentration, okay, for those waste products. So again, urine is concentrated. That's the end of the story here. The kidney functions to concentrate the filtrate by glomerular filtration. Failure leads to fatal dehydration. So if the kidney can't do what it needs, it just continually makes urine, okay, we would dehydrate very quickly. And water loss is very important because we sweat. Cooling the body, evaporative cooling, to keep our body temperature down is very important, but we can't just get rid of all our water because we would dehydrate. Okay, dehydration is quite an important pathological state. So we need to retain body water, okay, body fluids by kidney regulating the amount of fluid. Okay. Importantly, also, we absorb and retain valuable materials for use by other tissues. So sugars and amino acids, almost all sugars and amino acids get retained or reabsorbed in the body because those are valuable. Those are valuable building blocks. We can use those for energy or building other cellular products. So get rid of the waste, but keep the good resources. Waste goes out, good resources stay in. All right. So as we move into this next phase, this is what we call renal physiology. Okay, renal physiology focuses in on what are the processes by which our kidneys are doing some of these major functions. And so there's 3 major steps here in renal physiology that happen in the nephron. Okay, 3 major steps that happen in the nephron: filtration, reabsorption, and secretion. And again, if we've done the nephron drawing that will be assigned, we'll go into this in some detail. Each of these functions is associated with an arrow. The arrow is indicating traveling from the blood into the tubule or from the tubule back into the blood. Okay, and so the direction of these processes is important for understanding what's the overall function. All right, so again, just an overview: water and solute reabsorption. Reabsorption means keep the good stuff. Okay, we're reabsorbing it into the blood. The blood is what we keep in the body. okay? Filtration and secretion is going outward, going into the filtrate, okay, going into the tubule, okay? That happens mostly at the proximal and distal convoluted tubules. We'll talk about the locations all along. In the nephron loop, or the loop of Henle, we're primarily talking about water and ion movement, okay? So it's not so much we're moving wastes or nutrients, it's really just ions and water, right? So this green arrow indicates the location where filtration occurs. This is really the only place where filtration occurs in the glomerulus. We start with blood plasma exiting the blood vessel and crossing across the glomerular membrane, filtration, and becoming filtrate. The other two processes here, shown in the arrows all along the tubule, are reabsorption and secretion. And we will talk about those all along the tubule as well. So this is an important table, okay? This table shows the common components, the molecular components of plasma and urine, okay? In this column here on the left is the plasma, that's blood. And in the right, it's the urine, okay? These are the different ions at the top, metabolites and nutrients in the middle, and the waste products in the bottom, okay? So these ions, these are something that we can regulate as needed, okay? It's not like we need to keep all of them or get rid of all of them, but it's just as we need to adjust for sweating or blood loss or other changes in our diet. Sodium, potassium, chloride, and bicarbonate, we need a little bit, but it's not like we keep all of it, okay? So there's some in the blood, and there's some that's continually being released in the urine all the time. Okay, so there's an overall balance here. There's it just depends on the bodily needs. Metabolites and nutrients, you can see that the numbers are always big here on the left. Okay, we need these things in the blood, and therefore we do not get rid of them in the toilet because they're valuable to the body. So glucose, lipids, amino acids, proteins, you should see very little of these components in the blood, in the urine, because our body can use them and we can store them in other places. So close to zero, okay, some of these amino acids, we can get rid of some of them in the urine, but you should have no protein, you should have very little glucose coming out of your blood into the urine, okay, and very little lipid is coming out. But these are the normal numbers, okay? If you remember, we talked about insulin and normal blood sugars around 100. 100 is kind of the threshold of a high blood sugar, okay, or a relatively normal blood sugar. If you have numbers that are exceeding 100 milligrams per deciliter, then you're getting high blood sugar, okay? 130, 150, those are high blood sugars, although they can spike up to in the hundreds, okay? 300, 500, those are crazy numbers, but diabetics sometimes can have a really high blood glucose spike. Right. Okay, down in the bottom here, we want very little of these in the blood, and most of them, they're going to be concentrated in the urine. Okay, so urea, okay, relatively small amounts in the plasma because it's always present a little bit because we're getting rid of it. But compared to the urine, we have a huge concentration factor here. If we're going from this left column to the right column, we should see a very big concentration because these wastes are continually being pulled out of the blood and put into the urine. Okay, so anytime we're taking a measurement of blood and urine, we have different appreciation of these values. The goodies, we want more in the blood than in the urine. The baddies, we want more in the urine than in the blood. And these ions, it's give or take. It's about even depending on the situation. Okay. All right, filtration. Filtration is a function of hydrostatic pressure. That is the blood pressure pushing on the glomerulus. Okay, all those membranes are just a function of how much incoming blood pressure. Okay, occurs across the capillary walls. It's non-selective. Okay, I shouldn't say it's non-selective. It's size selective, but there's not individual molecules to say I want you versus you based on what type of molecule it is. It's really just a size exclusion. Okay, big molecules are excluded. Small molecules go right on through. Okay, so non-selective means that on the class of molecule. Okay, amino acids versus proteins versus lipids versus ions. Okay, filtration in some sites such as the liver, the pores are large. Plasma proteins can enter the interstitial fluids. Okay, so we talked about the differences in the different capillary types, the continuous versus the fenestrated versus the sinusoidal. Remember those three different categories of capillaries that we learned in the cardiovascular system? Okay, in the renal corpuscle, there are specialized membranes that restrict all circulating proteins, meaning we should not lose protein in the kidney. Okay, we should not lose protein in the kidney. If we're finding protein in your urine, that means that we have some compromised renal function. Okay, there's holes, there's injury, there's damage, but we should not see protein in the urine. So reabsorption and secretion, those are two functions that happen downstream from the glomerulus. Involves diffusion, osmosis, and some special mediation from protein channels and pumps. Okay, and so these are the four major types of processes whereby we are exchanging these molecules across the renal tubule. Okay, the first here is just simple diffusion. If you're a small molecule with no charge, you can diffuse the membrane. That's simple diffusion. Okay, here are these two proteins here, and these proteins or ions, not protein, these are small molecules and ions. The proteins are in purple, okay, and those are the membrane channels that are allowing the transport across the membrane. So this is called, all of these are called passive transport because we do not need ATP. Okay, passive transport means no ATP necessary. Okay, active transport, ATP, A for A, very simple to memorize. Passive transport, no ATP. But here we're talking about some specialized proteins, so we call this facilitated transport. Facilitated transport just means we need a protein, but we don't necessarily need ATP to work that protein. So facilitated diffusion usually means that we need a protein. In some cases, we need the motive force of some moving ion to help move the molecule. We'll talk more about that in a little bit. Active transport, of course, we require ATP. Where we usually, in this case, we are moving some molecule against its concentration gradient. It's like a fish trying to swim up a waterfall. Okay, normally, stuff just flows down a waterfall, right? That's just going from high to low. Okay, but something trying to go against gravity, well, that's going against a concentration gradient. Okay, and that would be something that we use for active transport. Again, just re-emphasizing what's the normal status of these ions in cells. Okay, cells are like a banana in the ocean. Okay, the cell is the banana. The cell is full of potassium. Okay, potassium is inside the cell, high concentrations. Outside the cell, lots of sodium chloride, like ocean water, okay, salt water. The net result on the charge is that there's an overall negative charge in most cells, okay, and that's why the grumpy face, okay, he's in a bad mood. All right, the pump that establishes this, of course, is the sodium-potassium ion exchange pump. We learned about this in the nervous system, and we can see the importance of this for maintaining those ions in neurons, but also this pump is in all cells that is continually establishing lots of sodium and chloride outside and lots of potassium inside. This pump consumes ATP. So there's a large amount of our cellular energy usage to generate this ion distribution. Okay, this distribution of sodium and chloride outside the cell and potassium inside the cell. That's the normal status of all cells. Why is that important? Well, we're going to learn why is it important to have sodium chloride out and potassium in, because all the other exchange pumps and channels use these gradients to move stuff. Okay, and they're very clever with how they do that. And this is the outworking of that previous figure. Lots of sodium and chloride outside the cell and lots of potassium or the other negative ions inside the cell. Now, how do we keep all of these transporters and protein pumps straight, okay, and organized? Well, this figure here is an attempt to kind of help you think through how these transporters work. One, how are they powered? and what are the mechanisms for the direction of movement. The arrow always represents the movement of the ion. Okay, the arrow tells you which direction it's passing. The thickness will tell you whether it's going down a concentration gradient. As you can see up here, you can see it's wide at the beginning and it's narrow at the end. That's the normal passage. So if you did not have a protein here, we would call this passive diffusion. But we need a protein channel, so that means it's usually an ion. The ion has a charge, therefore the charge molecule can't go through that neutral nonpolar lipid bilayer. Okay, so, but it's just going down its concentration gradient. No ATP is necessary. We're just going down the concentration gradient. Okay, it's just a channel-mediated passive transport. Okay, here and here. Now we go in, so let's talk about that. Everything flows downhill, easy peasy. Okay, the flow downhill process. Okay, and it's downhill because of this sodium-potassium pump. Let's not forget that pump is continuously working in the background. We're not talking about that pump right now. That pump has been set to the side and has established sodium's on the outside, potassium's on the inside. Now we're looking at these other transporters that are working next to the sodium-potassium pump. Okay, and this first category here, just flow downhill, just normal diffusion. Second major here is we call ride the salt. Okay, ride the salt, meaning given what the sodium potassium pump is doing in establishing these gradients, let's just allow, so let's say we have lots of potassium inside the cell, it normally wants to flow from inside out. It's going to ride its concentration gradient downhill, right? Well, if that wants to happen, that's energetically favorable. Could we do something else and kind of ride the salt, meaning it's already going that direction? Let's put something else along with it so that that sodium movement helps drive some other movement. And so it can be a co-transporter, okay, or it could be an exchange transporter. Notice that these arrows in the top are going the same direction, but these arrows here are going in opposite directions, okay? Now, at the top here, you can see that we're going from wide to narrow. This would be the activity of sodium or potassium moving down its concentration gradient, really driving this pump. And along the way, it's powering this less energetically favorable movement, okay? Because this one's going up its concentration gradient. You can see it starts narrow here and it goes to wide, okay? So that's called a co-transport system. Both molecules are moving in the same direction, but one is a favorable, one is a disfavorable. What does the favorable overcomes the disfavorable, and therefore we are achieving some work. This is an exchange pump, meaning the molecules just happen to be going in opposite directions. But think about, like, if you're going through like one of those circular doors in a fancy hotel, and you have some big guy that's like pushing through, okay, and he's the one that's turning the door. You have some little person that's on the outside, and they just kind of meekly walk through. They don't have to touch the door because the big guy walking through is generating the force to revolve the door. And the little person just kind of slowly walks at the right speed and it can just walk right through without touching the door. Okay, it doesn't have to do any work. That's the concept here. The big guy is moving down its concentration gradient, and the little person walking in is got a free ride. So ride the salt. Okay, the salt is already moving. Let's just follow and use some of that energy. Last category here is ATP forces it uphill. Okay, ATP forces uphill. This is what we call primary active transport versus secondary active transport. Primary active transport here, we always have to use ATP. We're hydrolyzing ATP into ADP, and the energy of that hydrolysis forces something against a concentration gradient. Okay, so it takes a little while to understand these different categories, but hopefully this helps begin the process of you memorizing the types of the categories in which we move materials across from the blood into the tubule or from the tubule into the blood. Okay, these are the types of transporters we'll be talking about in some detail. All right, so here's some of that in some of some words put to here. Specific substrates, the one direction, proteins vary, many types of carriers and proteins. Carriers proteins can be saturated, okay? This last point, number 5, there is a rate at which if you have too much sugar or too much salt or too much of anything, you can overcome the capacity for these transporters to actually effectively move them. And so you can go faster and faster and faster, move more and more solute across the membrane up to a point, and then it's saturated. You can no longer move enough across to keep up with the demand. And so sometimes that can happen in diabetes. You have way too much sugar in your blood, you're overcoming the amount of transport that the kidneys can do. And so glucose spills into the urine, and therefore if you see lots of urine, the glucose spills in the urine. If you see glucose in your urine, you know that your kidneys are not able to keep up with the reabsorption, and you probably have diabetes. There's probably some other reason. Why do we have glucose showing up in the urine? Right, so this is called the renal threshold, okay? It's the transport maximum, okay? In the plasma, it's the plasma concentration at which a specific amount of compound or ion begins to appear in the urine because transporters are saturated. Think about, I have high blood sugar, way too much sugar in my blood, and the other mechanisms are not able to remove the glucose fast enough. So my kidneys begin to spill glucose into the urine. It shouldn't go there. We should keep the glucose in the blood because that's valuable. But because we have way too much in the blood, it's like literally spilling over into the urine. Okay? The threshold for the amount of sugar has been reached in the plasma. Okay, the renal threshold. It's about 180 for the kidney for glucose. Okay? So here it is, 180 milligrams per deciliter is the threshold. If you have a blood sugar of 180, you're going to begin to see sugar going into your urine, and that's not good. Okay, this is a high blood sugar. Okay, it should be around 100. Okay, if you have well-regulated sugar, around 100, maybe 110 on a bad day, but diabetics regularly see it spike up into the 130s, the 150s, the 180s, up into the 200 or 300 if you have a diabetic is having a bad day. All righty. And so back in the cardiovascular system, we talked about hemoglobin A1C. Your A1C is a more long-term metric of how well your body is regulating the amount of glucose. And so a healthy A1C is around 6, okay, 6 or 7. You get up between 7 to 9, you're kind of pre-diabetic. And if you're above 9, you are diabetic. Okay, so the A1C value is measuring how much your blood has been exposed to glucose over the last month or two. Okay, it's a long-term assessment of how much blood sugar your body has been experiencing because it kind of attaches to this A1C molecule. Okay, so glycosuria, it means sugar in the urine. Okay, sugar in the urine is pathological. It's not good. We should not see sugar in the urine. That's why in the old days they used to taste the urine, gross, to see if somebody was diabetic, because if you had lots of sugar in the bloodstream, it'd spill into the urine. They could taste it, say, oh, this person struggling with diabetes. All right, amino acids. Okay, what's the threshold for amino acids? About 65 milligrams per deciliter. Okay, so it's lower than glucose. Okay, amino acid urea. This means amino acids in the urine. Okay, so this is the high threshold level above which, if you have this amount in your blood, it's going to spill into your kidneys and into your urine. So here is a kind of a cheat sheet on the molecules, and some of these molecules, these small ions, are reabsorbed. Some of them are secreted on average. These are no transport mechanism: urea, water, urobilinogen, and bilirubin. They're regulated in the other organs. Okay, these are wastes that are always coming out. These are miscellaneous that, again, it's variable. These metabolites are usually kept, okay, retained or reabsorbed. Okay, so osmolarity. Okay, osmolarity is the total number of solute particles per liter. Okay, this is an important definition for you to know. Okay, it's the total number of solute particles per liter. One moment here. Okay, I'm running out of time, so I'm going to lose my thing here. So body fluids have an osmotic concentration of about 300 milliosmoles. Okay, so 300 milliosmoles, memorize that. Okay, body fluids have an osmotic concentration of about 300 milliosmoles. Okay, this is the number of particles, ion particles in the solution. Okay, just count the number of ions. Okay, and you have an osmol. This number changes as we go through the nephron, and we'll see how much body fluids get concentrated, because we say in the urine it's around 1200 to 1400. Okay, do the math. That's about 4 times the amount that we see in our body fluids, 300 to 1400. Okay, we're doing the math. That's about 4 times the amount, 1200 to 1400. We have these other measuring things, these milliequivalents, okay, MEQ per liter, that you might also see. Okay, I'm going to skip over this because I don't care that you know all of these different values. So glomerular filtration is the first thing we need to look at. Okay, it happens in the filtration happens in the glomerulus, which is the capillaries are the glomerulus. The whole structure here encircled is the renal corpuscle, includes the Bowman's space. Okay, and there are three layers here that are used to pass materials through. Okay, first, so this is the activity of filtration. You can see this is the Bowman's space. Here is the glomerulus. If you were to zoom in here, you can see the interdigitated role of the podocytes as they're covering and wrapping around the capillary. You can see them here as well. Those create what we call a filtration slit. Okay, here's a cross section. On the left-hand side, this is the capillary. Here is the fenestrated capillary cells. This blue space is the dense layer. It's really kind of like a basement membrane. And then the purple, these are the interdigitated podocytes that you can see here. And through this little hole is called a filtration slit. The yellow arrow represents filtration from the blood into the Bowman's space, or into the tubule, the beginning of the tubule. So 3 things that it needs to pass through. Okay, the capillary wall, okay, the dense layer, and then the filtration slits. So first, the fenestrated capillary, they have pores about 60 to 100 nanometers in diameter. Okay, so here's the size exclusion. So if you have a large molecule like a red blood cell, for sure can't get through here. Also, most proteins are not getting through here because proteins are bigger than this diameter. Okay, but ions and small molecules, no problem. They are small. They can pass through. Okay, so that's number 1. The dense layer, okay, is more selective. Okay, it is positively charged. Therefore, small plasma proteins only, nutrients and ions. Okay, but it has a positive charge to it. That's important. Thirdly is the filtration slits. These are the finest filters. They have gaps of only 6 to 9 millimeters wide. Okay, so the difference between these two doesn't look, the artist has not drawn them to scale, but what you should appreciate is these slits are much smaller. Okay, only 6 to 9 millimeters prevent passage of most small plasma proteins. So if the dense layer was preventing some of them, these filtration slits prevent most of them. Okay, again, no proteins should be getting into the urine. So this explains why that is. It's a size exclusion principle. Okay, the big guys are going to stay in the blood. All right. So filtration pressure. Okay, again, all of this is driven by the pressure of blood pushing into the afferent arteriole. Okay. The balance of 2 forces here, you have the hydrostatic pressure. Okay, this is a push pressure, the fluid pressure, and then the colloid osmotic pressure. Okay, this is a pull pressure. This is the amount of pull of the molecules in the Bowman space. Okay, so 2 forces: the push of blood pressure and the pull of the colloid osmotic pressure. These are the same forces that we talked about in the capillary bed, so you should already be familiar with these. There's 2 spaces. There's the glomerulus, shown in pink, and there's the Bowman's capsule, shown in yellow. So we're filtering from the blood space into the Bowman space. Okay? And the big green arrow represents the overall filtration. So here we go. The blood pressure is exerting the glomerular hydrostatic pressure of about 50 millimeters of mercury. Okay, the blood colloid osmotic is pulling back about 25 millimeters of mercury. And this capsular hydrostatic pressure exerts a small pressure. There is some fluid in the capsule here. Okay, and so it is exerting some small push from the Bowman space back into the glomerulus, but it is small. So you do the math here, we have a total filtration pressure of about 10 millimeters of mercury, okay? 50 minus 25 minus 15 equals 10. That's the mathematics there, okay? We do all the math of the small arrows, we get the net result of the big green arrow. And that net result is outward, 10 millimeters of mercury, and most of that is due to the push of the blood pressure, okay? The hydrostatic pressure. So what does that result in? Okay, an amount of fluid per time, that is our GFR, okay, our glomerular filtration rate, okay, our glomerular filtration rate. This can be thought of as analogous to all the previous rates or metrics that we've seen. In the heart, we talked about cardiac output. In the lungs, we talked about respiratory minute volume, okay? So those are kind of two metrics, the kind of an amount per unit time. Kidney, same thing. We have an amount per unit time. So again, this is very analogous to cardiac output and respiratory minute volume. Okay, in this case, it is 125 mils per minute. Okay, that's the amount of filtrate that is being filtered per minute, 125 mils. Okay, about 10% of the fluid delivered to the kidneys leaves the bloodstream, enters the capsular space. So think about, for every 100 mils of blood that goes through the glomerulus, 10 mils is going to leave in the Bowman space. All right. Now, that's a theoretical measurement, okay? The GFR, and we can calculate that using a test, and this is called the creatinine clearance test. Okay, it is used to estimate GFR. However, that creatinine clearance test is laborious and it's tricky to do. So we use a more accurate GFR test, uses inulin. Inulin is a product of chicory root. Chicory root, it looks like kind of like a carrot, but it's not really a carrot. Anyways, you take this product of chicory root, okay, and this is the gold standard for estimating GFR. It's freely filtered, but not reabsorbed or secreted. So if you take it in, goes into the blood, gets absorbed, and it goes right into the urine and go right into the filtrate, and it stays in the filtrate. None should stay in the blood. Okay, so inulin should go right out of you. And so we can measure how much should we put in, how much do we get out, and then we can do the math and we can calculate what is our kidney's filtration rate, our glomerular filtration rate. Ready? So glomeruli can generate about 180 liters of filtrate per day. Guess what? Of that 180 liters, that's like, how many gallons is that? It's like 60 gallons, right? I don't know. I'll do the math. I don't know what liters to gallons is. It's about 2 to 1, right? But that's a lot. That's a lot of fluid. We don't pee that much. Do you pee 180 liters of urine per day? No. What do we pee? Only 1% of that. So we filter way more volume than we actually urinate. And that's because of this last sentence here. 99% of what we filter is completely reabsorbed. So we filter a lot and then we reabsorb almost all of it. Okay, that's the lesson here. So we can control GFR in a number of ways. There's autoregulation, that is the control of the vasoconstriction and vasodilation. We have hormonal regulation by the kidneys. We have autonomic regulation by the nervous system. Okay, so there's 3 ways here of regulating how much the glomerulus is filtering. They're going to go into these various mechanisms. I'm going to stop here because my computer is about to die. See you guys in class.